this Google Doc, and again, for those which were not here in the very early morning this morning, we took the registration information that you had provided and added this onto the Google Doc. And what we are doing right now is to see what additional changes have been made. Maybe some of the challenges have been overcome while we are together here all day. But I think there will also be additional challenges and additional opportunities that people would like to point out. Before we go there, it would be valuable to see if there are any leftover questions. I know during Jeff's session, there were quite a number of people that wanted to ask questions, but I think we also had fascinating talks, which used the entire set of 20 minutes that we had for each talk. So maybe, Jeff, if you want to see if there are any leftover questions, that would be wonderful. After we have answered those questions, let's go over to this document and look at what we got so far. Are there any questions regarding the regarding Jeff's session on ontologies, I think? Oh no, on anatomical mapping, sorry.
Yeah, there was one question from Ken Dunn, and you can, well, I can just read it. Simon, are you still available?
Yeah, I think they answered it, Jeff, in the chat window.
Yeah, I am here. I just turned off the video. Ken's question was really that in many of these optical platforms, the z speed is, or the speed in the z-axis is quite limiting, and we generally use things like piezo-driven z actuators to move very fast in z, and they have a limited range, normally between 100 and 250 microns. But in the case of this device, all the time is spent going in XY, and then the Z movement is achieved while the stage is homing back to the 0, position, the home position, and then it just starts again. So we've never had a problem with Z. You don't think of it as a normal optical sectioning device, which is a single picture and then in Z. This is XY and then Z. Does that make sense, Ken? Ken? Could you hear my answer, everyone else?
Yeah, I heard. So effectively you're taking like full two-dimensional images and then you go down in Z a little bit as you home back.
Yeah. The whole trick is the speed of the device comes from the ribboning, because it doesn't stop.
Yeah.
So the 30x speed advantage is from that, and also the way it scans. It uses a resonant 8,000 line pair resonance scanner, which is an inherently speedy scanner.
Yeah, there's some analogous technologies in the imaging mass spec side where continuous rastering allows you to acquire up to 300.
Yeah, we had a paper on using a imaging mass spec thing about in January this year. We were doing lipids, not proteins, and I was listening to your talk with interest because some of the problems you face are similar to the ones we face, but it was pretty high resolution. I think that's a tremendous opportunity, this high resolution scanning mass spec. I think we were at 1.5 to 2 microns in the mass spec, which is incredible, but it's the same problems.
Yeah. I'll look up that paper. I had a question in terms of
Zangawam Kemi.
Great.
Anyway, what's your question? Sorry.
I had a question about how many channels do you think would be able to do the ribbon scanning?
4. And it also does reflection, which is a really interesting twist, because you can combine the reflection information with one of the the regular fluorescent information channels, and generate essentially a histology image by combining those two channels together. The company we've been working on to build this thing actually are building a real-time pathology device for use in the clinic, which combines simple fluorescent stains and the reflection mode of this device to generate real-time pathology images. But it's 4 channels plus reflection, 765 nanometer reflection.
Were there any other questions for Simon?
Well, if any of you are interested in trying this out, let us know. We see that the questions being posed by HuBMAP and GoodMap and all these other mapping groups to be very applicable for this device, because it allows us to approach truly massive samples with very high resolution. And I look forward to hearing from any of you that are interested.
Thank you, Simon. Great work. I know that Bernard had to leave, but he posted on the challenges and opportunities side comments for Zarina, and I think they have interacted since then, so these are long comments. Are there any other questions regarding Zarina's talk or this Windbreak's talk? And that also had to run off. So maybe it's best to capture them here. But Zarina is still on, so if there are any more questions for Zarina's presentation, please let us know now.
So yeah, I've been answering and maybe some typos and things, but I'll be happy to have additional conversation. So feel free to shoot. And I'm still here. I turned off my camera because I got up. I couldn't sit anymore. Okay, I'm up now.
Yeah, it's a long day, but it's a wonderful.
Doing this.
So then coming over to challenges and opportunities. So again, this document is organized in a way that you have the 4 sections that we have been covering: metadata, ontologies, mapping and numerical construction, user interfaces, and you have challenges and opportunities for both of those. We also have references and links. So if you didn't have a chance yet to add your own presentations and papers there, please do now. Also in terms of links, if you want to link or send us to your GitHub repository or your online interfaces, please do add those links in here. In terms of challenges, we could go one by one, and we could do it in the same sequence as we had it here in the overall agenda. So I don't know if you want to take us through the metadata. I think, Sanjay, you...
One question, how were the first challenges put in? Because they were there already at the beginning of the meeting.
So all of these came out of the registration data sets. So as you might have seen, we gave you a very long form to complete. Some of you were very efficient and you just put in your name, first name, last name, project, and then as a reason for coming, I want an update. I think there are very efficient people among you.
I wanted to know who requires more guidance from the NIH.
I'll answer that. I'll answer it.
Okay.
Why isn't there a standard metadata table for a patient? Why does every funded group, every group funded by the NIH that actually has human patients generate their own metadata table?
I think Ajay already said that he will be happy to provide all that clarification. Ajay, are you on?
Yes, I am. Well, the reason is practical. We are too early in the system in our project to standardize on any of these things. Our approach is going to be, we are going to see what people are normally generating. We are going to bring those things together and make them standardized and look to see if there are specific use cases that these metadata will not cover. And once we agree that those use cases are important, that we need more metadata, we're going to go back and get more metadata. Now, of course, this is going to be an iterative process, which is why we have time at this point, at least, to start working towards converging on some of the metadata elements that we really need and which use cases those metadata are going to be useful for. And so articulating use cases, I think, is essential to get people to, go out of their way and add more metadata elements into any data set. That's my general answer.
In other words, there is a lack of guidance from NIH on standards for patient metadata.
That's editorializing there.
Well, no, we're developing it.
So I can point, I'm not speaking on behalf of NIH here, but I can point at least 2 pointers. One is that as part of BD2K, which is an NIH-supported program, there's a center, there's a CDR center that's been created at Stanford by Marcuson and others. And their goal is to provide better support for metadata. And as part of the CDR center, as far as I know, I haven't checked that in a while, but they have libraries so there are 2 things. They are a repository for metadata, and they also have standard metadata for projects that they store as part of the libraries. So I think that's one place where you could go for, if not guidance, at least examples. Another thing is that as part of the All of Us project, which is this large cohort that NIH is organizing and that connects both clinical and genomic data. All of Us has actually selected the format that they wanted to use for collecting the data, and they're going to use the HuBMAP version 5 clinical data model. And Oliver this morning talked about harmonizing across clinical data models. In this particular case, they picked HuBMAP over PicoNet over other possible common data models. So that might be an indication also of what kind of standard metadata for a patient. You're not interested necessarily in the clinical part of HuBMAP, but at least for the demographics, for these kind of things, there's going to be there's standard there that can be used. All right.
Okay. So what I'm still getting is that there's a lack of guidance from the NIH on the metadata standards. Because there would simply be a requirement that if you're studying humans, this is your.
I'm happy to tell you what metadata you want. It'll be useless.
In what sense?
Because without knowing what use cases we are going to put this to, asking me to specify or anyone to specify metadata is a waste of everybody's time.
I don't know. We have the problem with we can't decide on age being in years or months. We can't decide on body weight being in kilograms or pounds. We can't decide on sex or gender.
I mean, my general slide presenter, I don't think that's a problem. You're going to collect age and you're going to translate it. I mean, the fundamental problem is collecting stuff, not standardizing. We have plenty of standards to choose from. You map to these standards, you map it. So let me give you an example.
But the challenge comes when you have different types doing it different ways.
But let me give you a specific example. So Olivia raises HuBMAP, and then also mentioned the CDM for the Corey network, as well as there's an I2B2 network from the CPSAs. Okay. What we do in my shop is we pull data, clinical data across the board for 4.5 million patients, 1 billion lab tests, 200 million diagnoses, all this. Okay. We pull that into a data warehouse, and we generate all 3 of those formats for those 3 projects. Those 3 projects are run in my office. We generate an HuBMAP data warehouse, we generate an I2B2 data warehouse, and we generate a CDM, and it feeds into all of those projects. That comes from the data that we put together, and that works because we've done it in a decent atomic way that comes from the clinical data, and it's mapped. It's not a problem. You don't have to choose.
How many people did it take?
Most of that was done by 3 or 4 people over about a year. All of it was done by 3 people actually over a year.
Large data sets. So if you figure out how to map a particular source, you get 1,000 records mapped once you figure out how to do the first one.
Yeah, 4.5 million. They flow to all right.
So there's
4.5 million people.
high number of
flow to all of those different things. The point is to collect your data carefully, know what you have, And to me, all of the metadata and the ontologies and so forth are mapping issues, but you have to put them initially annotated to something.
Yeah.
In order to map.
You need to
And so
define
we're
spot.
doing that the way that we find that we need to do it, and then it's mapped and modified to meet the other one. So they're coming across, some of them have one, some of them do not, some of them have the CPT codes and some don't, and others, and we look at, and we do some 80-20 stuff. We have thousands of records that are not, there's thousands of elements that are not mapped, but they're a very long tail of something that we've done 90% of them with the ones that we have mapped. And those you can't query very well because they don't fit to a regular thing. We once in a while have investigators come along and say, do you have this? And we say, well, you can look at our local description. That comes with a local description with a local name inside the medical record, and our collaborator at Hopkins or Penn or whatever has a different local name for that, and then we go and tackle that one to Ajay's point. So I think that having a set of standards that we think are well managed and stable, reliably done, and being added to the points that Olivia were making, I think that's your choices are based on the ones that are going to be there over some number of years. And I think in that regard, CMS and in fact NIH have made some guidance on the clinical side of the world. They have specifically called out, they have specifically called out several others.
Arts norm and snow.
I don't think it really addresses the issue of this device works because there's a rigidly defined standard that everybody can design. There is no fuzziness in the specification for this device. How the GPS works.
And there's two major ones of those.
Yes.
Each of them are different.
And incompatible, and if I go across the border, they probably
You can write to both of them, and people have written software that you can write an application that will run on both of them, even though they're totally different.
I'm slightly more nuanced than Jonathan, and I'm with you with the exception of, I don't think we should access on age or on anything that's amenable to simple arithmetics, because as long as we know what the source is, if it's specified,
That was my point.
If it's specified in month, it can convert to years fairly easily. That's
You
when it's
can't,
not.
but it comes back to, yes, if I have a 10,000 record file and it's got age in months, it's easy to do a little bit of coding and get age in years. The problem is when you have a record that's all by itself. You have this you got a table with one line in it, and now somebody has to go in and figure out what the mapping is for it.
But it's that practice in the first place to have it's insufficiently defined data in the first place. So,
But I would say there should just be a standard table.
well, they wouldn't still resolve the problem of whether they would comply to the standard table. If they don't define their data elements correctly, they're in trouble.
So fortunately, you're both at dinner. Perfect for dinner. You're going to sit together, have a wonderful meal, and this hopefully does some missing. Richard also just added that he is agreeing that it's too early for standard, so you can read his text there. Thank you all for commenting on this text and adding to this document. This is gold on our end. Please do so. Also, I see Paul commented in many places. In the interest of time, we could go through the metadata challenges and opportunities. And I don't know if Sanjay wants to kind of steer us through these stormy waters. And then I think it's going over to the ontology, so I can moderate that. And then anatomical mapping. Jeff, if you could moderate that part. And then there's one last part, which is the user interface, which is Jim again. So if you can kind of make it through all these comments we got so far, that would be great. Sanjay, do you want to steer us through the metadata challenges? And
I thought we have discussed enough, but okay. So the metadata, as I'm looking at my screen, of course, the challenge is how to harmonize, So I agree that, some of the points that are being discussed, we want to really navigate the waters. And the comments seem to be saying that suggesting that one of the comments is balance between too little and too much information, which is very true because metadata can be thousands of elements, and we really need to understand for each organ systems, what do the domain experts and the users for which that data will be used is first essential to capture. And then the caveat should be that there always should be opportunity to, or permissions from the subjects to go back and get more. So I think that's critically important. And also be very careful where we don't try to go and get everything under the sun from the EHR or manual records. So that comment is posted, I don't know who posted it, but I think this is critical, and this will depend on each organ specific questions. And I believe efforts are already going between different domain experts to understand that at the use case level. The next bullet is ease of access, search, quality, and data manipulation. So some of these were, I think, presented in the talks today we heard on the metadata and some of the tools. Anybody has any comments or any suggestions over here? The user interface, I guess, is sort of ties up with the user interface session, I guess. So they are kind of connected.
And you could also go over to the opportunities for a moment so that we see all the options that exist to potentially deal with those challenges. Do you want me to go there?
Yeah, I don't see that on my screen.
All right, we have two opportunities, many challenges.
Yeah.
Well, two opportunities for metadata.
So I think the opportunities there would also be the tools at the interface level, right?
Yeah, but I think it's also wonderful to see that we had so many people from other efforts come in, because we do want to be aligning our efforts with other programs. I think we want win-win-win situations with other efforts that NIH and others are funding.
Right, and recycle, somebody showed a slide of recycling in ontologies. I think that's really important here, not to build from scratch if something already works.
One comment for metadata is that in our KPMP project, we will eventually use ontology to standardize metadata as well. I mean, currently we just began the study. We just focus on ontology development per se. But in the end, the idea is to also, once we identify the metadata and the structure them well, then we somehow align them into ontology. So make the tool become more like, in sync. Yeah.
Okay.
I just wanted to, we had raised this question when Bernard DeBono spoke about the metadata, the practical issues of getting the metadata associated with everything that comes in. And I think there's an opportunity there. I think it was actually just written as we were sitting here, that the opportunity to take the things that are standard, that are routinely done, make them standard. But I think there's, and that was just written up there, but then there's this other part of it, which is investing sufficiently in those folks that are writing and curating the metadata associated with the specimens. I think the idea that there is a solvable user interface that's going to get the folks that are focused on generating data to generate the associated metadata at the level that we want may not be realistic. I think that was his core message. We should heed that very carefully. And the question is, is that a challenge or an opportunity, in the sense that if we plan for it, it's an opportunity to have, metadata curators associated with data that are actively working right at the right time. You start them too early and we have the problem of they're collecting stuff that's not important. Start them too late, we've collected a lot of data that we don't have a piece of metadata for. I think that's a very key opportunity to get it right.
I'd like to share a story in support of that thought, in that in HuBMAP we had what we call legacy RNA-seq data sets that were there before our current team took over managing the project. And the way we collect metadata now for RNA-seq is different because it's evolved over the last 10 years and settling on more or less an ENCODE standard, we realized that a lot of the metadata that's captured as individual elements now was actually buried in the descriptive text in those 400 prior RNA-seq experiments. So we've had to go back in manually to pull that out and remap it. So some of that work is unavoidable. So planning for that with staff is a very good idea, in my opinion.
Yeah. And I think I have heard 4 to 5 people on Bernard's kind of side. I have heard how many on the KPMP side? How many people do you have that help with that part of the project?
Oh, you mean with the metadata?
Yeah.
Oh,
Data wrangler type.
yeah, everybody from TM's, the tissue interrogation sites are collecting the data within the standardized terms, and then the data visualization center has several folks who are actually talking to them to create the data. So at least 3 individuals from the data visualization center that Becky talked about, and each from the 6 tissue interrogation sites, there's like staff or project scientists from each of them who are working with them.
Yeah.
Maybe we should consider is we're collecting way too much data. His description was minimal, or one of the descriptions, minimal information required, minimal information solution. And then Becky had, there are 6,000 fields. Something's wrong.
Well, I mean, so you have to remember that KPMP is a clinical project.
6,000 boxes.
That one's different, though.
That's all the way down to the data.
That's the clinical data. That is the data itself.
Well, but that was everything, person all the way down to how the samples were handled. But is there any other place in science where you collect 6,000 data points for every line in the table?
Large Hadron Collider, I think.
Well, yeah, it's a lot of sequencing projects. When you publish it, you don't publish it.
We have million-dimensional data sets.
I know, but nobody publishes it.
Once it's right data, we can generate it. So this there's no defined boundary. I think it's a practical consideration.
Yeah, exactly.
I think 6,000 is too many. You're going to have a lot of
Depends how they're collected to you today. It depends how they're collected.
Not when every one of those items a person has to look at and make a call. It's not a vector of mass.
I agree with that. That depends how it's collected, right? So some of those things are collected from pulling them from clinical systems, and they come up and you get all this. Right. The ones that are entered by hand into a RedCap, you can't do that. Thousands are too many for you.
But I don't think the 6,000 that you're talking about were entered that way.
That's
no, but a big chunk were. And a big chunk were free text. So maybe part of the challenge here is figuring out, maybe we don't need provenance data.
Well, I don't know. That's another good dinner discussion. So let's go over to ontologies. So we do the same trick. We do challenges first. So Sanjay, thanks so much for bringing us through these stormy waters here. So let's go to ontologies just to incentivize everybody one more time to also look at those challenges. So we have lack of understanding of what an ontology is. Yes, some of the ontology interfaces we have seen today, I believe they are great for those which actually take good care of those ontologies and merge them and try to understand them and optimize them and use them. But they might not be valuable so much for patients, let's say, which are interested in a better treatment option, or for most of the researchers, which might just use the ontology for search and browsing. So I think it's really important to understand those different user groups, and Becky did a great job in showing us her user groups. So I really enjoyed that. Ability to effectively translate, integrate, and share access to many expertise domains that are needed for HuBMAP. Yes, that's another one. So I think you can read those much faster than I can read them to you, but there are many. So as you see, the ontologies have a lot of challenges. And if you have comments or additions to those, please add them now. This is your time. You can, of course, also come back, but What we will do with this Google Doc is to save it out probably on Monday next week, so that you have a little bit more time to add on. But then next Monday, we will take it off, create a kind of white paper out of it and share it back with all those which kindly registered. So if you have not registered yet, please do a minimal registration so that we can share that with you as well if you're interested. So I see much activity. This is good. Go do it. Then under opportunities, let's go down there. We have collaboration on ontologies of ontology. So just like we want an atlas of atlases, I think that's in the same spirit potentially. And yes, we don't want to make a new ontology, but want to use existing ones. But we have to figure out how to potentially add to ontologies.
There's an opportunity there that we can say, and I actually had a section there I forgot to show, which is that a number of these now have APIs that are sufficiently granular. So mine, for example, for every call in there, for every piece that interface is doing something, there's a call terms to terms, codes, concepts to codes, all of these things. I think the opportunity is not just finding ones that exist, but finding ones that have the APIs so the APIs can be easily constructed. I think that was one of the things that drives me very much towards UMLS is that the structure is consistent everywhere. And so that the APIs are very usable and doable. And then you can have many different interfaces for your different user constructs, and because the APIs are underlying it as opposed to them being tightly coupled. So I didn't present that, but what I showed is not tightly coupled to the interface at all. Every single piece of it is an API.
Yeah, and as others pointed out, most likely even the CCF, the main user will be a robot. I mean, will be somebody coming via an API. There might be some which use our CCF user interface that Leonard so lovingly designed and Bruce implemented, but many will just use the API and take what they want. But I could very well imagine that the CCF user interface could help those which are not database experts to construct the right queries to, for instance, linked data stores, which also not so many people are familiar with. I think most database experts out there, they are still thinking in the relational database mode. And so having the CCF show them here, this query you just did with this age range and that sex and that ethnicity and that organ part, this is how it is reflected as a SPARQL query. That could be very useful for many. All right, so CCF ontologies, opportunities. So please add to those if you have others. Can I put Jeffrey on the spot for the mapping and numerical construction part? So I can click here and go to the challenges. If I use the right mouse, I have to minimize.
Yeah, if we start with the challenges, you know, there are a number of them listed here. I'm not going to read through them all, but there was a lot of discussion around using the vascular system as a roadmap, if you will, for the human body. For all of those that were chiming in, does anybody want to continue this discussion right now? Some of these issues. I know one of the questions that I have that isn't totally clear is something that I brought up. I don't know if others have the same feeling, but it's clear to me how you would generate an average, basically a working average of the vasculature for kind of the larger structures. But as you get down to finer, 10 micron level capillaries, that would all, in my mind, just average out. And I don't know that I see the point of trying to go to those fine of resolutions in generating kind of an average framework. I don't know if anybody else.
So I tried to answer that, and maybe I'm not getting your question, but I think that if you try to imagine that you'll have 100 micron cube in any tissue, you will have a certain pattern that fits in that 100 micron cube, and most likely will contain a capillary or an edge, and there will be cells that are organized around it. And again, the pattern will depend on the tissue, and it's recognizable. And so my theory, and Kati took me up on this challenge, is that if the pathologist and recognize looking at the tissue section that contains 200 cells, what cells those are, what tissue it is, and whether it's normal or diseased, then it means that we have enough similarities where we shouldn't worry so much about really small level of variations. I don't know if it answers your question. I probably don't understand it.
Well, I guess I'm coming at it from the perspective of some of the more recent conversations we've had with Leonard and in how we precisely place, in our case, we're generating image data. You know, how we place image data within the context of the kidney. And it's our, at least our current perspective, obviously, we're open to changing our perspective, is that there's no need for us, you know, you reach a certain level of precision that you don't have that level of precision in terms of where we're collecting the tissue from in these kidney surgeries and where we're gathering tissue. So I don't know that it would be more than a rough guess for us to basically place tissue into a three-dimensional framework besides some of the landmarks that Sanjay pointed out. You can imagine doing it a little bit better, but I can't imagine getting down to the scale of even 100 microns in how that would function practically.
This is
that. I mean, again, if you look at, first of all, you have a pattern that's already known. So we are not reinventing the pattern of the kidney. Everybody has an idea from many anatomical pathological studies, right? So what you're trying to do is basically just to anchor the genomics, proteomics, and things to certain types of cells in known locations, right?
Yeah, so from our perspective, the way we envision that happening is, you know, we can give you an image and we can segment it and say, okay, this part is medulla, this part's inner cortex, this part's outer cortex. This comes from the 5th pyramidal structure in the lower pole. And then that's probably enough information. I don't know what other, we're obviously willing to collect any other information that's needed, but because we have the ability to through pathology and through specific antibodies for cell types, we can give all of that information for all of the data within our images. I'm not sure, and it's probably just me not understanding, I'm not sure what having an additional framework on top of that brings to the table.
I think it's a multi-scale model issue. So there is a model at the lowest scale of a vessel in the pathology slide, and it doesn't matter whether it's north, south, east, or west at that moment within the context of that slide, and it's not on a mathematical GPS that can get up to the largest level in a model that will cover all humans. That's not the issue. And I think Zarina presented that. It's a series of multi-scale models. So you have a model of what it means that the vasculature is going through this tissue. At that lowest level, you have that. You can't connect that for all the reasons that you described back to the aorta in a complete way, either semantically or, well, semantically maybe, but certainly not spatially through a series of connections where you have everything at every step. So you have these multi-scale models, and I think the point is to make sure that they're connected enough at the key stepping points between multi-scale models that you can function and drive down and up to what you want to see. But I think that's the critical piece here. Yes, they are disconnected. They have to be disconnected. They're at different scales. We don't even have the information to connect from the largest to the smallest scale because you need all the tissue. We're not going to do that.
There's the fact that nature is highly parallel.
Yeah.
Which is why you can have a humongous variability in number of nephrons, but when you get down to them, they're all going to have very similar structures. So maybe we ought to be mapping out what the typical nephron is rather than the typical kidney.
Yeah, and I think we saw that.
It's a space filling algorithm after that, saying how do you connect enough nephrons to get the job done, with some variability across the organ, and that's what gets interesting, right? But rather than saying, can I place this specific nephron number 27,432 in patient 1 versus patient 2, when that's a meaningless question in the sense you can't connect one person's nephron number, whatever I just said, I can't even remember it now. meaningless comparison. That's more meaningful.
I think we can also imagine the fact that we are going to map certain things extremely well based on previous known information, new information that we get and everything. And the rest we are going to assume that are similar based on knowing the pattern, right?
Yeah, that's multi-scale modeling. That's exactly what it is. It's a pattern at each different scale. You get it onto that pattern and then you can function. It's on the map, right?
Yes.
That's what I think. If you get down to the wrong scale, trying to use something as a coordinate won't make sense because it's more stochastic than anything else that will be conserved. You can't map Ganymede onto Callista. They're both rocky moons of Jupiter. They both have crusts and things like that, but you're not going to map them on one another. You just map them individually. And then compare what you can, the size, diameter, mass, how spherical they are, and things like that. But
remember that Sanjay showed us that there are 4 or 5 different types of endothelial cells in a fairly small area of the kidney, right? And we actually know that in that area of the kidney you have certain structures, so you can map those cells back to the areas and the structures that you know they come from. And then you know what cells are next to them, again, because this is known for many, many years. And now we are just seeing a even more resolution. We are seeing what they make and how they react to being in a neighborhood.
I mean, what is important in kidney, at least, is take the functional unit of the kidney and have an understanding of its connectivity to the neighboring structures, rather than assigning, very exact sort of GPS-like coordinates. What is important is harmonizing the cell type identities with the structures, the regions, and surrounding areas. I think that's where the strength is. And once we understand that among different individuals, and when we do validation studies, you'll be able to see if what you have generated from your sampling of limited area, how well it reflects on the rest of the organ, right? So, that's something we can't really predict because we don't really have that much data yet. So, we have to understand what the biological variability is going to be at that level, and some of these things may not even matter. We may be able to learn which are the regions of the kidney which it matters the most. So, I think, and the cells will carry inherently structural information among them and their positional relationships. So if you take a principal cell or a proximal tubule cell, its cell signature will sort of give you an idea what could be its neighboring space. So, and when all that information is together, with some 3D imaging tools and technology we heard, I think that, to me, if we can accomplish that, we would have a really good understanding, at least at the atlas at that level, which is probably will be a big leap, I think, so.
Yeah, so the functional unit in almost every organ contains a capillary, in brain, in kidney, and everything you want to look. So there is a reason, because this is where oxygen, nutrient signals come from the rest of the body. So that's why I thought it's kind of the thread that connects everything. And as Sanjay said, yeah, once you characterize and you understand the functional unit of every organ, after that, the rest is kind of just putting it together. We saw this also from Bernard, right?
Yeah, I think having a kind of network understanding and knowing how important the surrounding environments are in which these cells live is very, very interesting. And in the interest of time, let us go over to the CCF ontologies opportunities. Let's see what got added there. And we already went there before, sorry for this. So let's go to CCF mapping and construction. And that's what we just had. All right, never mind. So now we want opportunities for mapping and numerical construction. Okay, there are a few. So we have Fantastic Voyage, which was mentioned before Serena's talk came on.
I said yes. I don't know who said, but yes.
So,
I think that's also part of what we would like the interface to look at. I mean, think about how exciting it would be for people to be able to do that, to either their own or anything, have augmented reality in which you could go inside the body or inside the blood vessels or whatever.
Yeah, yeah.
That's what Becky was talking about.
And again, you can also read what is already here. If you have other comments which you would like to add, please do so.
I want to make one comment, which I made there in writing, but sort of maybe just reemphasize. I come to biology from my physicist perspective, and I believe we are more similar than we are different. So having a lot of anxiety about how different we are going to be or small variations, I think it's focusing on the wrong thing. I think we have to start from stuff that is really common, because all these are solutions that have been refined over thousands of years, millions of years, whatever. So we all work. We can talk, we can look at each other, we can think and things like that. So focusing on the commonality and then discover the normal variation and the abnormal variations would be very interesting.
Yeah. All right, last but not least, CCF user interface challenges. Not too many compared to the other challenges we just saw. But I think we are all still struggling to understand what these new data sets will actually do in terms of clinical impact and in terms of research opportunities. And I think just like the Human Genome Project produced data and much of the usage of that data was not foreseeable, I think also HuBMAP might produce data which we only find out later how useful that is for all kinds of applications that we can't yet envision. But we have all started now to look at different personas, to have in-depth interviews. And again, thanks go to Sanjay and Jeffrey for having us meet with them every other week and really truly trying to understand their world and what's feasible or not on their world. And same also to Xin Lin. I think this very close collaboration is absolutely necessary. Tim, did you have any comments? This was the one that you chaired, this section. So then maybe just an invitation for you all to add other challenges that you see and opportunities. And if we go over to opportunities for CCF user interfaces, we also have quite a number of them that I think the most important part is also to empower us all within HuBMAP to understand 9 different organs, or now 10 different organs, 25 different assays. I think just within HuBMAP, this is already kind of overwhelming. And then, of course, communicating that to the outside world is even more challenging, because they are not like we in day-long meetings about all of this, but they have maybe 5 minutes of time to understand it if they are patient.
We do need to think more about interpretation as opposed to, for instance, I don't think it adds much to an end user to tell them that that was mass spec data in the interface. I think it's much more useful to tell them, to ask them what fatty acid you want to find, or what protein you want to find. So I don't know, I mean, obviously we have to have a way to click through to a particular assay type, but really most users are going to come in with more of a biological point of view.
I agree.
Ask for a gene and get mass spec data and in situ and.
Yeah, I mean, part of what I was showing with the ontologist was exposing what's going on in the equipment. In the synonomy, for example, I wasn't imagining that you actually show someone the synonomy. The imagination is you type something in, and any of those set of things are already understood by the machine to do that same concept, and it can pass through. Right? And in that same vein, and so I think the elegance of the interface is dependent on all of that intelligence underneath it. And that's where we can really gain something special.
And I think actually building on both those points, users aren't really caring perhaps about it being mass spec unless there is mass spectrometry.
No, and then you have to drill down and all the rest.
And then also they don't necessarily care about a particular cyclin or a particular case 7. What they really care about is those approaches that tell you that you're doing cell cycling.
Yeah.
So you're searching for an activity or a behavior And then it should suggest which proteins map onto that, and then you go search for that data. You know, I envision you want to search for a certain cell behavior or activity or process, and then figure out what should be visualized. And if you could do that, actually, that would be immense use for a lot of biological community, because you're spending years just learning what proteins are the standard proteins to image for some particular thing. You learn that CD31 is a gold standard marker for a blood vessel.
They haven't done that yet in single cell sequence data. They haven't done that yet in single cell sequence data. As far as I know, when they show a cluster, that's just numeric values of genes with no biological context. And I don't think they've yet gone into those clusters and say, what is unique about this gene set like you would in a microarray.
They're starting to. Like,
They're starting to, but those clusters are not showing up.
I think there's some pathway-oriented ones that we've seen, but they're early. I think they're early.
Early, yeah.
It's coming, but it's.
Oh, yeah, it will come. We would have to, otherwise.
Well, it's
you're not
impossible.
getting
It's
anything
not
valuable.
just someday it will happen. We're working on this after.
Well, the Human Protein Atlas actually has that sort of designation where it's grouped into all of these things.
It's along integrated.
Yeah, I mean, some of the problem with the ontologies is that stuff where it's embedded, it's not. This gene is of this class, of this type. All those classifications are in there. You get them for free, right? Because they've already been classified, right? So you do need that base level information, gets you on the map. They've already been classified. You want to say you want to come in and see the ones of this particular pathway. Well, that's known. You say this pathway, it translates to that's all this set, right? And we'll now look for that set. So managing all those pieces, I think, is the real power. That's why that's a particular example why I worry about trying to create. You can't do that. That's many years of curation that has created those relationships, and they all exist.
Mechanical Turk will help, and data, literature mining, things like that, because people who characterize these proteins. So if we could connect that, that would be enormous.
They're welcome. I mean, Go and some of the others have them connected all the way from pathways to diseases to, stuff is unbelievably connected.
Very good. This is a great discussion, and I hope it got all captured on these two devices we have here. So again, all the presentations will be made available. And before I give Jeff and Sanjay the last word, I would like to thank all of you one more time for coming in. I think we have quite a number still left after a long day of presentations. So thank you for that. Please do add to that document by Monday, let's say noon, and then we will take it offline and work on it more and make it available as a white paper. Also, thanks to NIH for encouraging us to have this workshop together. I think it was extremely
providing guidance.
Yeah, yes. But also support, of course. And so we all here will have a nice dinner. I hope you have a nice dinner with your families.
Cook it.
Again, there is another workshop planned. Actually, that one is organized by Rahul's team. And that workshop takes place in Boston. But I think there's also a virtual component again. It's on June 27th in the morning. So if you liked what you saw today and you want to do that again for 3 hours, please do come back on June 27 in the morning and just watch out for the HuBMAP newsletters. I don't know if those are shared outside of HuBMAP, but there's also the consortium website, and there's now a Twitter account for HuBMAP, which also emanates this kind of information. Again, if you have not uploaded your final slides, please do so. If you would like to connect to anyone, we have not shared email addresses of participants. Let us know if you saw somebody on chat and you really want to connect. Just come back to us. I think we want these networks. And going forward, we hope to really create a community of people that care about common coordinate frameworks, and that would, over time, really grow something that none of us could do alone. So thank you again for coming in. Jeffrey and Sanjay, do you want to add to that?
No, thank you. Kudos to you and your team to really pull this through in a virtual manner, and pleasure working with Jeff on this, and thanks to everyone from my side.
Thank you, Sanjay.
Yeah, just to reiterate that, thanks to all the speakers, and particularly thanks to Kati and her team for doing much of the heavy lifting. It's I'm sure it was a lot of work for everyone, but especially for her team. You did a great job.
Yeah, and Liesl and Alan and Leonard and Matt, they're all here, so maybe you could give them a round of applause.
Thanks, everybody.
Thanks.
Thank you.